Hello dear students, welcome back to Biovidyalaya. I hope you all are doing very well. Today in this lecture video we are going to discuss about phloem loading and unloading in plants. That means food transportation in plants. In our previous lecture video we discussed about phloem structure. This is the continuation of that lecture video. So please try to watch that video first. I will give the link in the end screen. And also don't forget to watch this video till the end. Then only you can understand this concept. So without further delay, let's get into today's class. In our previous lecture videos, we discussed that phloem is a conducting tissue that will conduct food, and main conducting cells present in phloem is CO2. Okay. So here translocation of organic solutes such as sucrose takes place through CO2 elements of phloem from supplying. Supplying means source to conception end. Conception end means sink. Okay. So phloem conduct and distribute sugar made in the leaves. That means that is made in the source to grow in tissues and organs that cannot carry out photosynthesis. That means sink. Okay. So here you can see that mature leaves are called source that will synthesize their own food, and that will transport that food to sink. Sink means that may be flowers, stem, roots or developing leaves. Okay. So here source is transporting food to sink via CO2 elements, that means phloem. Okay. So this sink includes shoot and root apex, flower buds and developing fruit and seed. Okay. So source makes sugar. Source makes sugar. That means mature leaves. And sink. Sink may be flowers, stem, root or developing leaves that uses sugar. Source makes sugar and sink uses sugar. So food is transported from source to sink via phloem sieve tube. Okay. This transportation of sugar is not unidirectional, it is multidirectional. It can move upward, it can move downward, it can also move in lateral direction. That's why it is multidirectional. Okay. And transfer of sugar from mesophyll cells. We know that photosynthesis is occurring in mesophyll cells. That's why mesophyll cell means that is source. That's why transfer of food from mesophyll cells or source to sieve element is called the phloem loading. Okay. So transfer of sugar from source to sieve elements is called the phloem loading. Okay. And transfer of sugar from sieve elements to receiver cells of conception end, that means sink, that is called the phloem unloading. Phloem unloading. That means transfer of sugar from sieve elements to sink is called the phloem unloading. So transfer of sugar from source to sieve element is called the phloem loading, and transfer of sugar from sieve elements to sink is called the phloem unloading. So the movement of food or the transportation of food through this phloem is based on mass flow hypothesis, that is also known as pressure flow hypothesis. Okay. If you don't know what is osmosis, then this concept will be little bit difficult to understand. So I suggest you to please watch our latest video, diffusion versus osmosis. So please try to listen here carefully. So we know that food is synthesized in mesophyll cells. That means sugar. Sugar is synthesized in mesophyll cells. Then it is transported to C elements via companion cells. Here you can see that this is mesophyll cells. Mesophyll cells. And this is companion cell. So sugar that is synthesized in mesophyll cell will move to this C element via this companion cell. That's why what will happen? Solute, that means sugar concentration in this C element will increase. That's why osmotic gradient will create. Because of this osmotic gradient, water will move from xylem. This is xylem. From xylem to this C element. So, hydrostatic pressure or the turgor pressure in this C element will increase. Turgor pressure will increase in this C element. Okay. So, this pressure that will cause sap to move out of this C element through this pores. That means here C plate is present. Through this C plate, this sap will move. That can move downward or upward. On the other end, zinc is present. Here you can see that zinc is present. This zinc will take this sugar. That's why what will happen? Here sugar concentration, that means solute concentration, will decrease. That's why here osmotic gradient will create. Because of this osmotic gradient, water will move from phloem to xylem and this process will continue. So here now there is a difference in pressure between source phloem and sink phloem, isn't it? And this causes this difference in this pressure that will causes this phloem sap to flow, and this process is known as pressure flow mechanism or mass flow mechanism. Okay, I think this is clear for you. Next we can do a detailed discussion of phloem loading and unloading. First is phloem loading. There are two pathways for phloem loading. That means the initial short distance transport of photosynthate from mesophyll cells to phloem can take place through apoplastic or symplastic pathway. Okay. I think you all know what is apoplast and symplast. We already discussed that in one of our lecture video. If you didn't watch that video, then please do watch. So, symplast means it is the transport of sugar through plasmodesmatal connection through cytoplasm via plasmodesmatal connection. Here plasmodesmata is present. Through this plasmodesmata, sugar will transport. Okay, this is cytoplasm. And apoplast means it is the space between cell wall and plasma membrane. If this is cell wall, this one is cell wall, and this is plasma membrane, and the space between cell wall and plasma membrane is called the apoplast. So transport of sugar through this space between cell wall and plasma membrane is called the apoplastic pathway. Okay. And this Symplast pathway is passive process, and this Apoplast pathway is active process. That means that need energy. And both these pathways begin in the same way. As we know that cytoplasm of the most of the plant cells are interconnected via plasmodesmata. So first from mesophyll cells, sugar will move to the bundle sheath cell. From mesophyll cells, sugar will move to bundle sheath cell. Okay. Through plasmodesmata. From this bundle sheath cells, sugar will move to phloem parenchyma through plasmodesmata. Okay. From there, this pathway will differ. That will occur apoplastically or symplastically. Okay. So in apoplastic loading, sugar initially get transported through the symplast till phloem parenchyma, after which they enter the apoplast quite near the C elements companion cell complex just prior to loading. Okay. And this is very important, the route of phloem uploading. That means phloem loading is very important. Sometimes you will get part C question based on this. So the route of phloem uploading is mesophyll cells to bundle sheath cells, from bundle sheath cells to phloem parenchyma, from phloem parenchyma to companion cells, and finally to C element. Okay, or sieve 2. This is very important. You have to remember this. First we can discuss about apoplastic phloem loading. Before that, it is better to know about different types of companion cells. So which are the different types of companion cells? Ordinary companion cells. Ordinary companion cells. Transfer cells. And intermediatory cells. Intermediatory cells. So ordinary companion cell, transfer cells, and intermediatory cells are the different types of companion cells. We are not doing the detailed discussion, but you have to remember that in this ordinary companion cells and transfer cells, only few plasmodesmata is present. That's why it is following apoplast pathway. But in this intermediatory cell, many plasmodesmata are present. That's why it is following Symplast pathway. Okay. So if the companion cell is ordinary companion cell, then the phloem loading is apoplast pathway. If it is intermediatory cell, then it is Symplast pathway. Okay. So let's see how this apoplast is working. Apoplastic pathway is working. Okay. We know that mesophyll cell is the source. Here only photosynthesis is occurring. That's why here sugar is synthesizing. And this sugar will transported to this is sucrose. Here blue color one is sucrose. That means sugar. This sugar will transported to bundle sheath cell via plasmodesmata. This is plasmodesmata. And from this bundle sheath cell, this will move to phloem parenchyma cells via plasmodesmata. And here you have to remember that this is companion cell, but this companion cell is ordinary companion cell. That's why only few plasmodesmata is present. Okay. So here what will happen? Until this phloem parenchyma, it is for sugar transport is symplastic transport. After that, it is following apoplast transport, because this sugar will enter into apoplast area. That means the space between plasma membrane and the cell wall. And here what will happen? Here ATPase is present. This is ATPase. H plus ATPase. This H plus ATPase will pump H plus into this apoplastic area. Here in this SCCC complex, ATPase is present in the plasma membrane, and this ATPase will pump H plus into the apoplastic area. Okay, that's why what will happen? Proton gradient, that means the H plus concentration will increase in this apoplastic area. So, because of this proton gradient, H plus will move down its concentration gradient to this SCCC complex via this sucrose H plus symporter along with sucrose. Okay, so here H plus here sucrose H plus symporter is present. This yellow color one is the sucrose H plus symporter. Okay, because of the high concentration of proton in this apoplastic area, this H plus, that means proton will flow to SCCC complex, okay, through this sucrose H plus symporter along with sucrose molecule. Okay, and now this sucrose is inside this SCCC complex, then it will follow Symplast pathway. Okay, I think this is clear for you. So the sugar from apoplast are loaded into SCCC complex, that means C element companion cell complex, by an active ATP dependent mechanism that involves a plasma membrane bound sucrose H plus symporter. It is an active transport, that means that need energy, and energy for uptake is provided by the plasma membrane ATPase proton pump. Here you can see the proton pump that will pump proton from these companion cells to apoplastic area. So ATPase pumps proton out of the cell into the apoplast. A high proton concentration in the apoplast generates a proton gradient whose energy is used to drive the transport of sucrose into the symplast of SCCC complex through a sucrose H plus symporter. Okay, I think this is clear for you. Next we can see Symplast pathway. So Symplastic pathway involves the participation of special cells present near the phloem called the intermediatory cell. So the type of companion cell present in the symplastic phloem loading is intermediatory cells. We know that intermediatory cells have many plasmodesmata, so they can follow Symplast pathway. These cells have many plasmodesmatal connections, and this sugar is moving through this plasmodesmata by simple diffusion. That's why there is a chance of reverse flow. But how it is prevented? This reverse flow is prevented. That is explained by the polymer trapping model. A polymer trapping model has been proposed to explain the symplastic phloem loading of sugars. Okay. So, polymer trapping model of symplastic loading. As we know that sucrose is synthesized in mesophyll cells, from there it will diffuse to bundle sheath cell, from bundle sheath cell to phloem parenchyma. But here in this picture that is not shown, and it will finally reach to sieve element via this intermediary cell. Okay. But here in this intermediary cell, sucrose is converted to oligosaccharide like raffinose and stachyose. So here sucrose is converted to, some sucrose is converted to raffinose and stachyose from sucrose and galactose. Okay. And here now sucrose and raffinose concentration is more. You have to remember that all the sucrose are not converting, but some of the sucrose are converting. That is for maintaining the concentration gradient. So now the concentration of raffinose and stachyose is more here as compared to sieve element. That's why it can move to sieve element via plasmodesmata, and here the plasmodesmata is wider. Okay, so it is this big molecules can able to move through this plasmodesmata. Okay, both sucrose and raffinose can move to sieve element. So here as compared to bundle sheath cell, here sucrose concentration is less, isn't it? Because some sucrose will convert to raffinose and some will move to sieve element. That's why here sucrose concentration is less. That's why it will not go back to bundle sheath cell. And but because of the higher concentration of sucrose in the bundle sheath cells or the mesophyll cells, sucrose from mesophyll cell will continuously come to this intermediary cell. Okay, and but this raffinose concentration is more in this intermediary cell, but it will not diffuse to bundle sheath cell because here this plasmodesmata is not wider. That's why this big molecule are not able to go to the bundle sheath cell. Okay, I think this is clear for you. So sucrose is synthesized in mesophyll cells, diffuses from the source to the bundle sheath cells through plasmodesmatal connection, finally reaches into the sieve elements via the intermediary cell. In intermediary cell, sucrose is converted into series of oligosaccharides like raffinose or stachyose from sucrose and galactose. This results in an increase in the size of carbohydrate molecule, and these polymers cannot get back into the bundle sheath cells and are thus trapped, but at the same time can diffuse readily into the sieve cells due to larger concentration in the intermediary cells. Since sucrose gets continuously utilized in the intermediary cells, concentration gradient is maintained so that there is a flow of sucrose from mesophyll. Okay, and in the zinc cells, this raffinose can be converted to sucrose. Okay, I think this is clear for you. Next is Florium Unloading. There are 3 steps in Florium Unloading: sieve element unloading, short distance transport, and storage and metabolism. In sieve element unloading, sugars leave sieve element of zinc tissue. And in short distance transports, sugar transported to cell in zinc by short distance pathway. And in storage and metabolism, sugars are stored or metabolized in zinc cells. Okay, and like Florium loading, Florium unloading may take place by Symplastic or Apoplastic route. That is depending upon the nature of zinc tissue. As I told you that symplastic and apoplastic pathways depending upon the nature of sink tissue. We know that growing tissues are metabolically very active. Okay, that's why in young dicotyledons like those of beetroot and tobacco, the phloem unloading and short distance transport are entirely symplastic. But in storage organs like fruit and seeds may show an apoplastic step in addition to the symplastic one. So in symplastic phloem unloading, transport sugars such as sucrose move through plasmodesmata to the sink cells. In the sink cells, sucrose can be metabolized in the cytosol or the vacuole before being stored or entering metabolic pathways associated with growth of the tissue. Okay. So here you can see that here sucrose is there. This is sieve element and this is zinc. Okay. So here sucrose is moving to zinc via plasmodesmata. There is no need to cross any plasma membrane. That's why it is moving by simple diffusion. This is a passive process. There is no need of energy. Okay. So here sucrose is moving to zinc cell via plasmodesmata. Okay. When phloem unloading is apoplastic, the transfer sugar can be partially metabolized in the apoplast, that is, get hydrolyzed into glucose and fructose in the apoplast by the enzyme invertase, or it can cross apoplast unchanged. So here in this picture you can see that sucrose is entering into the apoplast area. This is apoplast area, and from here they can directly go to zinc via this plasma membrane. So here they have to cross the plasma membrane. So crossing this plasma membrane, they need the help of transporter. That's why it need energy. Here sometimes sucrose will convert to glucose and fructose in the apoplast. Then it will go to zinc cell. Here also for crossing plasma membrane, that they need transporters. That's why it is energy consuming process. That is active process. Apoplast pathway is active process that need energy. Okay. I think this is clear for you. This video might be little bit lengthy because I included many information in this video. That's why please try to watch this video till the end without skipping, and also don't forget to share this video with your friends. And please try to watch all the videos related to xylem and phloem, because in our next video we are going to discuss about some previous year question based on xylem and phloem structure and function. Okay, so here I am winding up this video. If you like this video and find it helpful, then please give me a thumbs up, share this video with your friends, and also don't forget to subscribe this channel. Your suggestions are very important for me, so please comment your suggestions in the below comment section. Keep learning. Thank you.